Ok, d'accord.
Je ne dis pas.
Il
Ouais, il faut être sachin. Alors, si ça passe, on en paiera que la moitié. Si ça passe pas, ben, on en paiera entièrement.
What, sorry?
Yeah, maybe you want me to click again? Yeah,
Yeah,
that should be fine. Okay.
So now we can start?
Yeah.
Okay. Hi. Thank you. Yeah, thank you so much for the invite, and thank you for being here to listen to me talk for a bit. So as Antoine was saying, one of the things that I've been doing for the last few years with Oscar, Pierre, and Jerome has been trying to understand a bit how can we identify local adaptation in quantitative traits, and specifically under this problem of structured populations. So I'm bringing these two images here. So on the left side, you have a 9-spine stickleback, and on the right side, you have what I expect is the range of these fish. And I brought these images because I wanted to bring attention to 3 characteristics of metapopulations, which I think are quite inevitable when we're thinking about quantitative traits and quantitative genetics, these types of populations that we'd study there. So the first thing is that, well, it's quite unlikely that a fish from northern Alaska will have the same migration rate or same relatedness to the population in, I don't know, northern Canada than it would to Quebec or it would to Scandinavia. So by that, what I'm saying is that there is likely structure in this population, and that the this structure is not homogeneous throughout the whole range. The other thing is that because we have this huge range and because there are microhabitats throughout this range, we should also expect that there are some traits that we'll be interested in studying that will have variations that are doing better in some environments than others. So there's likely some type of local adaptation happening in some of the traits we'll be interested in. And finally, that despite this huge range, there's likely some parts of the habitat that is similar throughout the whole range, or aspects of the historical contingency of these fish or physiology and so on, that makes it that certain variations of phenotypes we'll be studying will do better throughout the entire spread. So there is some kind of global adaptation. So I use these 2 terms, global and local adaptation, which I'm borrowing from a paper from Sam Yeaman. And I will come back and refer to them a few times throughout this talk. And when I say global adaptation, I'm talking about a scenario that we have a trait that the fitness function or throughout the metapopulation is very similar. So here I have these individuals in red and blue depicting 2 different populations. So this red population and blue population, and they're experiencing the same fitness optimum. And while with local adaptation, I'm talking about a scenario where we have that the individuals from the red population experience one fitness optimum, while the individuals in the blue experience another one. So they have different habitats. So I brought this example, which Antoine very kindly allowed me to be exploring, which is with these 9-spine stickleback. And I showed you this huge range that they have. But even if we were to just look at Scandinavia and these Finland and Sweden populations, knowing that these fish occupy both marine and pond water, so this fresh water and this salty water, we would expect that there are some selective pressures that differ between these 2 habitats. And then also Antoine noticed that, and his collaborators noticed that the head of these fish, depending on where they came from, looked very different. So the question would be, like, is there a difference in the type of food that they're eating, or maybe some kind of predator or some kind of behavioral difference that is created from this habitat that they're occupying? But before we can start creating these adaptive narratives, we need to figure out if this phenotype and this variation in the phenotype we're studying, it has like high enough heritability for adaptation to be occurring. So what I'm saying with this is that it's important to do some standardization of the environmental variants. And I brought the more popular cousin of the 9-spine stickleback, which is the 3-spine stickleback. And this work by McCarran and Benachet, where they noticed that in the wild, in the wild, these freshwater individuals and the marine individuals had very different morphologies. But once they brought them to the lab and reared the offspring of this wild individual in the lab, they noticed that the variation that they saw before between like the marine and the freshwater really decreased, showing that a lot of the variation that they were observing came from environmental effect. So, but of course, these guys knew what they were doing, and they reared their fish in captivity, and they still saw a big difference between the marine and pond fish in terms of the head morphology. So this is what these graphs are showing, that there is some differences. So the question is, are we seeing local adaptation? Naively, we can think that, well, we have a narrative, we have this difference in habitat, we also have a difference in this trait that we're studying, so it is likely local adaptation. But we'll, as we all know, if you have finite populations, which is the case of these fish, of course, and if you have some restriction in gene flow, you should expect that drift alone could drive divergence between subpopulations. So the question that I was interested in the last few years is how should we identify this local adaptation in these structured populations? Because there is this divergence that will inevitably occur due to drift in these finite populations, the first thing we should do is find out how much divergence do we expect under neutrality. And this is what we do when we measure FST on the neutral, on neutral loci. And when we do this measure, we're asking ourselves how much divergence is explained by drift. And then we can compare this amount of divergence that we observed with the FST value with the divergence we observe in our trait. And to do this, the classic way is doing a QST-FST test, which was formalized by Spitze in '93. And QST is an analogous of FST. So I probably you all know, but FST, when we measure FST, what we're measuring is a ratio of the heterozygosity that we observed between populations. So in this case, between that population, this population, and the total heterozygosity. So it's a comparison of how much divergence occurred. And QST is also a ratio, and it's comparing how much divergence occurred, but at the phenotypic level or the additive genetic variance level. So to measure QST, we're going to have to measure additive genetic variance between beams. I should have a little, and the total additive genetic variance in your population, which is the between and the within together. I'm going to be using VB and VW as additive genetic variance throughout this whole talk. So I brought this example in graylings. I realize now that all the examples I have are on fish. I don't really study fish very much, but I guess they're a great study system. So yeah, with when you measure QST-FST, if you have that QST is larger than FST, which is an example here on these two populations of graylings with the growth rates and with yolk sac volume, for example, it means that the divergence that we observe at the trait level is greater than that we would expect by drift alone. So it would be an indication that there's possibly selection happening and possibly local adaptation. But if you have a scenario, for example, that FST is greater than QST, for example, like incubation time or length time termination, it would be an indication that you have less divergence than that you would expect by drift. So it could be an indication of global adaptation. And finally, when you have that FST is about the same as the QST of your trait, so hatching length is an example here, drift alone could explain the divergence that we observe. But there are some issues with QST-FST. There are many reviews that talk about all potential issues with this method. Here, because I'm interested in population structure, I will focus on the issues related to measuring these variances, and specifically with the how we measure additive genetic variance between beams, because I'm interested in structure. So normally, the way we get these variances is through a linear mixed model. Here's what I'm showing you here. So you have Z is your trait. the trait value, the mu is the mean value of your metapopulation, AI is the individual level random effect, AP the subpopulation level random effect, and the epsilon is your residual. And the way we get these variances is that the additive variance within comes from the variance of this individual level random effect, and the VB comes from the population level random effect. The thing is that there's an assumption with QST that this population level random effect, the distribution is a normal distribution and an isotropic normal distribution. So this comes from this identity matrix here. Oh, sorry, I should be pointing with this. It comes from the identity matrix here on this equation. And it's basically, in biology terms, it's saying that all pairs of populations are just as related to each other. So we are making an assumption that you have something like an island model. And just jumping the gun a bit is what we're also making an assumption that the demes are about the same size and so on. So if you have something that is quite different, so for example, this stepping stone population structure, you could expect that there is an issue there. So this was the very first thing that I did actually in my PhD, was to figure out if this assumption was indeed a problem or not. And I did that by I simulated 500 neutral traits in different population structures, and I measured QST-FST on these of these traits, well, QST of the trait and FST of the whole genome. And because they are neutrally evolving traits, when I get the p-value of these QST-FST tests, I would expect, when I get the whole distribution, I would expect something like a uniform distribution with about 5% false positive rate. So 5% of these tests should be showing that there is some kind of local adaptation happening. So I did this with the island model population structure, and as we expect, it works quite well. You have this quite uniform distribution. But then I also did with other population structures, for example, the stepping stone. And when you have this divergence from island model, you can see that this is not a uniform distribution. It's quite L-shaped, meaning that you have a really high false positive rates. So I wasn't the first, despite doing these tests, I wasn't the first one to notice that there could be an issue there. Actually, Ovaskainen and collaborators not only did notice that there was an issue there, they also proposed a solution for this problem. And in fact, the work that I mentioned by Antoine, they also use this method that they developed. So I will give a quick and simple explanation of what they did, how their method works. So this is our population level random effects that I showed earlier. So I had told you that they use, usually you would have this identity matrix here. And their solution was, instead of having an identity matrix, we'll have a co-ancestry matrix. So this, in very few words, I'll talk about it a bit later, but in this co-ancestry matrix is going to give you a co-ancestry value for all pairs of population. So it'll give you a better description of the demography of this metapopulation. And here, instead of the VB, you might have noticed that you have a G matrix. This is an ancestral G matrix. So a G matrix is a variance, covariance, additive genetic variance and covariance matrix. So because their test is a multivariate test, so you can be looking at multiple traits at once. So yeah, so then they, instead of using p-values, they have this S statistics, which so you would have your observed pattern. So this is the AP. But you also have these random draws from the normal that I showed you, the normal distribution that I showed you earlier with the G matrix and the theta p. And S, as I written here, is the probability that a random generated pattern, so the AR, would show equal or greater deviation from neutrality than what we observe, this AP. Yeah, so then you have the S value. If you have an S about 0.5, then drift alone can explain the divergence we observe. Around 0, it indicates global adaptation. Around 1, it indicates local adaptation. So they also have an implementation of their method, which is called drift cell. And in this paper that they present the implementation, they also suggest a way of calculating theta p. So this coancestry matrix, which is using an admixture F model. I also did some tests on these, and other people also tested how to measure theta p. And what I found is that it's a more, there's some issues with the accuracy of AFM, and the method of moments seem to be more accurate. So in the test that I'm showing you, instead of using our AFM, I used the method of moments because it gave better results. So yeah, so the same test that I did with QSTFST with the exact same traits and same evolutionary history and so on, I did with drift cell. So we are not expecting, I'm going to show you the result, but we're not expecting now to see a uniform distribution. So drift cell uses the Bayesian approach. So you have like an MCNC, and we're going to get the last however many iterations and get a mean. So because we're getting a mean of a distribution, we expect to see a normal. So it's going to be normally shaped. This is not weird, but the thing is that I don't really know, don't know if anyone does, what to expect exactly of this distribution. So what I did was the way that I referred to false positives was using the author's description. If something is larger than 0.2, sorry, if something is under 0.2, I would call it global adaptation. If something is over, I'll call it over 0.8, I'll call it local adaptation. So of course, drift cell does quite well on the island model, which is expected, but on this very different structure on the stepping stone, it had quite high false positive rates. And yeah, so because of this, I reached out to Otto Avaskainen and Karunen, who were part of the group of people that developed this method. And well, I showed like, these are just a few of the many structures and different setups that I tested the method, and we discussed a bit. And then we were like, okay, maybe we can suggest an improvement to their method. So using many of their ideas, maybe we can do something about this. So this is what we did together. And this was it is one of the chapters of my PhD, hopefully. And yeah, so before I get to exactly what the method is, I'm going to give you some background on the quantgen that we use to come up with this method. So let's say that we have this ancestral population here. I don't know the first rate of that right here. This is the ancestral population. And then with time, you have that this ancestral population diverges into two or into current demes. So quantitative genetic theory says that if you have a measure of this the ancestral adaptogenic variance, and you have a good measure of demography, so here I'm saying FST, you should be able to derive the additive genetic variance within demes for your neutral trait that evolved in this period. Mm-hmm. And similarly, using this equation, and similarly, there's an equation that you could derive the additive genetic variance between demes for this neutral trait you're studying. So if you think about it a bit, it makes sense. If you know where you started in terms of variance, and you have the demography pattern, so how they diverge between each other, you should be able to know how much difference is there between the two demes and how much difference is there within each deme. But I said that you needed a good measure of demography. And if you have a scenario that you have two demes, they're exactly the same size and so on, then FST would do the job, but FST is a single value. So if you have a complex pattern, so you have had many demes of different sizes, and the relatedness between them is different, either because of history or because of how much migration you have between them, then FST is not sufficient to describe these complex structures. But as I showed you earlier, Avaskainen and collaborators had used this matrix instead, which is this theta matrix, which is a coancestry matrix. So we also followed their steps and used this matrix. So for this first equation, so these two equations that I showed you earlier are the basis of the entire method. So for the first equation, this between variance equation, we're going to use a matrix that we're referred to as theta p. And theta p describes the coancestry between pairs of demes. So with itself, a population itself will have high coancestry, of course, and a population in another, for example, this A population here that has that is very far away will have lower demography. So for this equation, instead of using FSD, we're now going to be talking about theta p. And so for the within population, the additive genetic variance within, we're going to use this M matrix, which is a block diagonal matrix. So while theta p is number of populations by number of populations in terms of dimensions, this M matrix is going to be number of total individuals by number of total individuals in terms of dimensions, but it is a block diagonal matrix. So we are looking within each deme, what is the coancestry of pairs of individuals? And instead of using the 1 minus FSD, we will use this M matrix. So again, going back to that linear model I showed you earlier, now we have the covariance is explained by this M matrix for the individual level random effect, and the for the population level random effect, you have this theta p matrix. And I'm showing you here that I'm using a VA comma W and a VA comma B. So this is still the ancestral additive genetic variance, but one comes from this within equation and the other comes from this between equation. And because this is kind of a detail, but because we're talking about these are matrices, at the end we're going to be talking about the covariance between and the covariance within populations. Instead of a single value, you have this sigma B and sigma W. But yeah, so given that you have some trait values, which I'm using the Z there, and you have a measure of coancestry for your subpopulations and within your populations, we should be able to infer using those equations the ancestral variances. So we're going to infer using this first equation, VAB, and this other equation, VAW. And given the theory, we expect that under neutrality, these two estimates should be about the same value. So these are the ancestral variances. This is going to be our neutral hypothesis. And our test statistics is going to be the log ratio of these two. So in the last little bit, I've been trying to write the implementation of this method, which is, a lot of it is just using BRMS. So if you're familiar with our package BRMS, this is going to be very simple to use. I hope so. So a lot of what I just said has been published already, and on this publication, we also did some preliminary tests that I'm going to show you now. So again, this is the same set of traits that I tested QST-FST and drift cell. I also tested our method, which is this log AV. I'm going to refer to it here as log AV. And it seems to be well calibrated, that you have around 5% false positive rates on both this island model and also the stepping stone structure. And also on the many other structures that we tested so far, it seems to be well calibrated. Just that we tested some other structures, which I'm showing you here, but it's well calibrated throughout. Also, for the preliminary tests, I also did some tests with selection. For these first tests with selection that I'm showing you, we have a scenario that the metapopulation is spread across two fitness optima. So basically, you can think of it as half of the population of the metapopulation, so half of the subpopulations are in one habitat, and the other half is in another habitat. And what will change between the scenarios is the width of this fitness function. So wider fitness functions will mean weaker selection, narrower, stronger selection. So now I'm showing you the log ratios as opposed to showing you the p-value distribution that I did before. And because these are scenarios of local adaptation, we expect the selective scenario should be all over 0, so up here. And of course, the neutral scenarios, we expect to cross 0. So again, we did with 500 traits that were evolving, but in this case with selection. So the neutral case crosses 0 as we'd expect. And the cases with selection, for the most part, they do. We see a consistent, we consistently identify selection, except for this case here, the island model with very weak selection. And well, I didn't really know if it was because, so in this case, the island model has 8 deems, while with stepping stone has 20. So I didn't know if it was the number of subpopulations that made our method have a weaker power, or if it was at the combination of that and the very weak selection. What I'm showing you on the left set of graphs is the trait distribution. So on the y-axis, you have the trait values. On the x-axis, you have the population. And you can see that strong selection, so omega 10, you have very different traits on different habitats, while on the omega 50, it's very similar, the traits. And since we have the many populations of stepping stones, you can really see a gradient of these traits. So, well, because of that, I was like, oh, I think it's worth making some thorough tests and finding out the limits of our method, and also finding out when does the method lose power. So I looked at quite a few parameters. I'm showing you some of them. So I looked at population structure. We now kept the number of populations the same throughout the different structure, but we also played with number of population across the different structures. We looked at different selection parameters. So for example, as I said, the fitness with the funk. So if it's very wide, it's weak and so on. And the optimum difference. So if we had many habitats in these scenarios, and if you have that the habitats are very similar or very different, how does this impact power? We also looked at this one that I thought was kind of cool and I was excited about, which is a selection to migration correlation. So in this case here, you have the population 1 and 2 are very related, 2 and 3 are related too, but 1 and 18 would be quite far apart. But you can see that in this scenario, you don't have a strong correlation between selection and migration, because, for example, population 4 will be experiencing this peak-like habitat, same to population 10, while population 2 and 3 will be in this lower habitat. Anyway, just to say that you can think about these different cases of correlation between selection and migration. Yeah, and well, we also looked at FST and number of populations. So for these results, because we're looking at so many things at once, the way that I thought would be easier to digest with using ROC curves. So we're going to be looking at true positive rates on the y-axis and false positive rate on the x-axis. So one important thing is that the x-axis is in log scale, because we want to look at this bit here that is under 0.05. We want to focus on it. But we're going to be looking at the 3 different methods that I mentioned. So lava is log AV, the one that we developed, RIFCell and QSTFST. So they will be in different colors, these curves for the different methods. And we're also looking at the different widths of fitness function, so how strong selection is, which are going to be in different shapes of the line. And we're also looking at global adaptation and local adaptation, but one is weak, meaning that those habitats are very similar, and one is strong, meaning that the habitats are quite different. So first, let's look at island model, which is a simple case. And as expected, seems like all methods do pretty similarly, like they do pretty well in all cases. Of course, if you have weaker selection, is going to have lower power. But we did this with quite a few different structures, but I'm not going to bore you with all of these. But we also looked at stepping stones. And in this case, it makes sense to be talking about the correlation between selection and relatedness. And we're looking at 2 different ones. I'm showing you 2 different ones. One is Klein, which is this one. So you can think that there is a strong correlation between selection and migration. And the other one, I call it Sign, which there's no correlation between selection and migration, or weak. correlation. So again, we're going to be looking at global and local adaptation. For the local adaptation is the one that makes sense to talk about the correlation between selection and relatedness. Yeah, this is what I said earlier. So, okay. So now we start seeing that the methods act different. In blue, maybe it's a little small, but in blue, so this one is QSTFST, and this yellow and red is lava and drift cell. And I want to draw your attention to these two cases, which I thought was really interesting. That it looks like QSTFST is doing super well in this client scenario. So the scenario that you have high correlation between selection and migration. And while in the sign case, when you don't have correlation, drift cell and lava, which correct for local adaptation, for population structure, are doing much better. So this is not too unexpected. There are previous work that talked about this. But if you have a method that doesn't correct for population structure, then the population structure, so the neutral divergence will be observed as if it's selective divergence. So it will add to the power of it, but actually a lot of what it's picking up is the neutral divergence between these traits that would happen even if there's no selection. And it becomes like, I think it's an evidence that this is what's happening, because when you break this correlation in this very similar scenario, QSTFST really loses power. We also did quite a few other tests on like common gardens and sampling, on how it impacts the power of lava and the other methods as well. So my idea was kind of create some kind of guideline of how many individuals you should be considering, how like phenotyped individuals, and how many subpopulations would ideally be sampled, and so on. I can talk to you about it later at another time. I'm skipping for now, but let's go back to the 9-spine stickleback. So as I said, Antoine and his collaborators had used drift cell to measure this, and when they used drift cell, please correct me if I miswrote, but they noticed that the 3 first principal components, so they did they had these markers on the fish, many markers, I think something like 50 something, and also they measured head size, and they noticed that these 3 first components and head size seem to have to be under local adaptation according to drift cell, especially when you considered habitat as the cause of it. And so I wanted to see if lava got similar results. So the method that we came up with, if it got similar results. So I applied the same data that they did with drift cell on lava, and we're using 2 different models, one with habitat, this one, and one without habitat. So the one without habitat, I will explain the one with habitat, maybe it's easier to understand, but the one with habitat means that if you correct for habitat, you still see this a log ratio difference. So this, and the other one would be without considering it. So what it means is that if we see a positive result with this guy, but not here, it would indicate that likely habitat is playing the role on the local adaptation that we would observe. If we see a result here, but not there, it means that there's something else playing a role. So the first component here that also they observed evidence for local adaptation with drift cell, we also did. So in this graph, what I'm showing you is the posterior distribution of the log ratio. And as expected, because we were looking for local adaptation, it's over 0. This dark patch is the confidence interval. And what I'm showing you is that on the 2 models, we do see local adaptation. So what I think this points to is that habitat might play a role in the first component, but there is something else also playing a role. So there's another selective pressure that is unrelated to habitat playing a role. And so the head size component 1 and component 3 pointed to being impacted by habitat. So habitat being a selective pressure that is causing local adaptation. But there's something else playing a role as well, because we see signals on component 7, 1 again, as I mentioned, and 12 when you take out a habitat. So we saw kind of different results between lava and drift cell, which I think is cool because it means that I didn't fully waste my time. Yeah, so I tried to give some conclusions. So none of these are very new, but I'll share anyways. The first one is that population structure seems to matter quite a lot and is something that we should consider. The other one is that it seems that it's quite challenging to detect local adaptation because, well, we do have high power when you have super strong selection, but maybe when a lot of the selection we might be measuring is not that strong, so it would be tough to see. And detecting global adaptation is even harder. Yeah, and as I said, we are writing a package that hopefully will be usable soon. Yeah, thanks so much for paying attention to this, and thank you for inviting me. And also, of course, thanks to my supervisors and to my group. And yeah, that's it.